Okay, good afternoon, students. Let us continue with our previous class, wherein which we were studying about the structure, reproduction, and significance of sargassum. We studied that the sargassum is one of marine aquatic algae, which belongs to the subdivision Phaeophyta, that is brown algae, because of the pigment system and the cell wall component, the type of reproduction, and the kind of flagellation they have. And this is one of an algae which has been widely distributed all over the world, especially in the tropical and temperate oceans. You can see them. There are around 300 species, and about 10 species of sargassum has been found in our ocean also, both in east, west, as well as in south ocean. Coastal region you can come across in India. And this is commonly called as Gulf weed. And the distinguishing characteristic feature of this particular organism is the kind of thallus, macrothallus it has got, which looks like the plant, the angiosperm, bushy plant it looks like. And this is one of a weed, aquatic, marine, or a seaweed, which is one of the biggest seaweed bloom which you could come across in Gulf of Mexico. And we also studied about its habitat, as I had mentioned in my previous class also. This is one of algae which looks like a angiosperm plant, bushy plant it looks, and most of the species are found to be floating. So as you could see in this particular image, they are floating form, and some of the species, say for example, Sargassum vulgare is being found attached to the substratum with the help of attachment disc. That also we have studied. And also you could see the thallus. The thallus, if you could see, you can find the stem-like structure that's called as central axis, and you could see the branches. The branches here, they are called as laterals, and the leaves-like structure here, it is called as primary lateral, secondary lateral, etc. And you could see something fruit-like structures which could be compared to that of a fruit, but it is the vesicle or the air bladders which helps in the process of flotation or for the process of buoyancy. So, and also we studied in detail about the thallus organization, that is how the plant body, how actually the structure of this particular sargassum is, that is the morphology of this particular algae we studied in detail, wherein which we studied that this particular sargassum, as I had mentioned before, it is in the form of the floating algae, or it is an algae which is being found attached onto the substratum deep inside the ocean, or it could be found floating. The flotation is due to the presence of a special structures, as I had mentioned, due to the presence of a special structures called as vesicle or air bladder. If you see the plant or an algae macroscope, it is a macroscopic algae, macro algae. It has got a central axis, that is the central axis you could find in these images, the central axis which we could compare to that of a stem of an angiosperm plant, and it produces branches. Here the branches are called as laterals or lateral branches. They are dorsiventrally flat. They are dorsiventrally flat, and the phyllotaxy, phyllotaxy is the mode of arrangement. It is either, if it is a central axis, it is spirally arranged, and the number of the leaf per node can vary from 2 to 4. That is, the phyllotaxy is 2 to 4, and it is arranged in whorls. And these particular laterals can also give the branches. That is called as primary branch. They can produce primary branches. See, this is the main branch, and you could find some branches of limited growth, that is our primary laterals, which are called as leaf-like structures. And there are also certain branches from the main branch. These are called as branches of unlimited growth, or the primary branches. See, these are primary branches. The primary branches can also give rise to secondary branches. Okay, so now the leaf-like structure is called as lateral, and the leaf-like structures from its axils, they produce certain vesicle or a ball-like structure filled with air, that's called as air bladder. The main function of these air bladder, as I had mentioned, is buoyancy. They help in the process of flotation. Another thing is this particular plant, they undergo the reproduction by fragmentation as well as by sexual reproduction. Most of the species, they undergoes the process of fragmentation. Few of them can undergo a process of sexual reproduction, where the plants or these particular algae can be either a monoecious or a dioecious. That is, the plant, in one thallus you can find both the sex organ, that is called as monoecious. Dioecious, the male and the female thallus are completely different, that's called as dioecious, where you can find this gametangia. So here we are not going to call it as a gametangia. Here we are going to call it as conceptacles, male conceptacles and female conceptacles. Where you could find this conceptacles? From the lateral branches, there arises axillary branches. These are axillary branches which arise from the axil of the lateral branch. From this, these particular branches are called as receptacles. They are called as receptacles. You can find receptacles here. These receptacles, in these receptacles are embedded with some cup-like structures or flask-like structures have been embedded. Those flask-like structures are called as, or the cavities which you can find in these receptacles, they are called as conceptacles. So remember, receptacles, receptacular branches has got receptacles. Inside these receptacles, you can find what? Conceptacles. What are these conceptacles? These are the structures which produces male and female gametangia respectively, or male and female gametes respectively. Female gametangia or female conceptacles produces oospores or oospheres. It's called as oospheres. The male conceptacle produces Antheridium. Antheridium in turn produces the male gametes. Okay, they can be called as antherozoids. Okay, and also we studied the anatomy of this particular algae. We studied the anatomy, the transverse section of the lateral or the leaf, where you can find upper and the lower the region, that is meristrodum. It's called as meristrodum, upper and the lower meristrodum. And this meristrodum is being covered over by a cuticle. Just beneath this cuticle, you can find the elongated cells made up. They are palisade layer. They are palisade layer. This is these elongated cells. They are palisade layers. They have a plastid in them. They have a pigment system, that is chlorophyll A. Remember, chlorophyll A, chlorophyll C is the dominant photosynthetic pigment you can find. Along with that, you can find fucoxanthin, that is the xanthophylls which you can find. So you can find abundant number of the pigment inside them, and also you can find the reserved food materials inside this particular layer. And just beneath this layer, palisade layer, you can find the cortical region, where these cells or these particular layer, in this particular layer, you can find the cells which are comparatively thicker, and the main function of this cortical cells is mechanical support. And in the innermost layer, you can find medulla, where you can find the cells which are loosely arranged. In some of the species which are in the form of flotation, that is where they float, those species, here medulla region completely it is hollow. You may not see any cells at all. The main function of this particular medulla, which is lacking the cells in them, they are filled with air, hence they help in the process of buoyancy. So that is the anatomy which we studied. And if you see the transverse section of the central axis or the main axis, again you can find the barrel shaped cells in the outermost layer, that is called as meristrodum, and you can find the cortical layer, and in the central there is a medulla, and they are thick walled. If you see the anatomy of this particular algae, you could see the differentiation of the labors, that is each cell perform the different particular function. So for example, in the lateral, that is leaf-like structure, there are elongated cells where we could compare the cells with that of a palisade parenchyma of our angiosperm plant. And there are certain hypodermal cells. They are thick walled, where you can compare this particular cells or a tissues to that of the sclerenchymatous tissue, which mainly help in the process of mechanical support when compared to that of an angiosperm we are talking about. Medulla region, they are loosely arranged cells where you can compare this to that of a spongy parenchyma. The main function of the spongy parenchyma is they are loosely arranged cells. They have air cavities inside them, where they have, in this air cavities, the air has been filled. Because of this, the plant can float. Hence, we can tell that there is a great evolution in this particular thallus organization of this Sargassum, where we could compare this morphology with that of as well as the anatomy with that of a higher terrestrial plants, angiosperm plants. And also there are we have also studied the anatomy of the receptacles also. So these are the receptacles where you can find the conceptacles inside them. So this is one of the anatomy, the transfer section of the receptacle, where they carry sterile conceptacles. Sterile conceptacles are the one wherein which no sex organs or the gametes have been formed. Such conceptacles are called as sterile conceptacles, where these conceptacles, they are flask-like structures, and from this flask-like structure, sterile hair-like structures, multicellular hair-like structures arises. Those hair-like structures are called as paraphysis. These main function of the sterile conceptacle is they keep the receptacle moist by absorbing by producing certain mucilaginous substance. During the process of unfavorable condition, these sterile conceptacle make these receptacles are kept moist because of the presence of the sterile conceptacles. So till this we studied in our last class. Now let us proceed with the reproduction in case of Sargassum. So as I was mentioning before also, this Sargassum asexual method of reproduction, that is by the formation of any kind of spores, is completely lacking. Asexual spore formation is completely lacking. The most of the species of Sargassum reproduces usually by the process of vegetative method, that is by the process of fragmentation. This is one of the common method of reproduction you can find in all the species of Sargassum. However, sexual reproduction is also being observed, where you can find the type of sexual reproduction. Here it is oogamous, that is by the formation of Oogonium, Oogonium carrying the egg inside them. So because of the formation of this Oogonium, the method of reproduction is called as oogamous, and sex organs are produced in a flask shaped cavities called as conceptacles. So as I was mentioning, so for example, if this is a thallus of Sargassum, you know, this is one of the branch which I have shown. So if it has got an axis, it produces some lateral branches. Those can be compared to that of a leaves, or some phycologists call it as a blade also. And some of the blades or the lateral branches, they get modified into the vesicles or air bladders. And from the axis of this blade, you can find some special structures or special branches which arises. Those are called as receptacles. And if you observe that receptacles carefully under microscope, inside these receptacles, on the surface of these receptacles, you can find some cavities. Some cavities can be observed on these receptacles. Those cavities are called as conceptacles. These conceptacles are of two kinds. As I was mentioning, female conceptacles, male conceptacles, and also I mentioned that some of the species are monoecious. That is, in a single receptacular branch, you can come across both male and female conceptacles. But female in one of the in single conceptacle, both Antheridium and Oogonium may not be formed. Remember, always female conceptacle is being produced in a separate conceptacle. Both male and female reproductive structures is not formed in a single conceptacle. That we have to remember in monoecious species. If they are dioecious, so it is in case of dioecious species, what happened? The female plant or the female thallus is different from that of a male thallus. Hence, the female thallus produces only the female conceptacle. Male thallus produces only the male conceptacles. So the sargassum reproduces sexually by oogamous, that is by the formation of oogonium, and they produces a special branches called as receptacles. The surface of this receptacles embeds the structures called as conceptacles, which forms the cavities inside this receptacles. And we have two different kinds of conceptacle, female conceptacle and male conceptacles. Now, let us study in detail about each conceptacles. So, the female conceptacle is our oogonium. So, here, let us study how the oogonium develops and what is the structure, how actually the structure of oogonium or a female conceptacle is. Say, for example, on the receptacular branch, some cavities forms, some cavities are produced. Imagine a receptacular branch. On the surface of this receptacular branch, if you take a transverse section of such receptacles, you can find some hole-like structures. Those are called as cavities. Okay, at the lower region of this cavity, what happens? Some of the cells at the wall of this cavity, they become distinct. How they will become distinct? The nucleus, they will have a prominent nucleus. See, you can see there are certain cells which are getting distinguished from that of other cells, where those cells become, the nucleus become distinct, and they have a dense cytoplasm, where you can find the cytoplasm which is dense. They have a reserved food material inside them, and such cells which have been distinguished, they are called as oogonial initials, where this oogonial initials have been produced inside the cavities, where these cavities are formed on the receptacular branches. Some cavities are formed at the lower or at the inner side of this cavity wall. You can find some of the cells which are getting distinguished. That is, they will have a prominent nucleus and dense cytoplasm. Those cells are called as oogonial initials. Later, what happens to this oogonial initials? They undergo one transverse division. The oogonial initial is undergoing one transverse division. Lower cells is called as stalk cell, and the upper cell is called as a oogonial cell. So, this oogonial initial, it undergoes one transverse division, where the lower cell is called as stalk cell and the upper cell is called as a oogonial cell. And remember, the sargassum is one of a diploid plant. It is a diploid algae. That is, they have 2n number of chromosome. So this is a diploid plant. Oh, sorry, algae. So what happens later? So this nucleus, which is diploid in a oogonial cell, they undergoes one meiotic division. Remember, the nucleus undergoes meiotic division followed by mitotic division, which leads to the formation of 8 nucleus. So here, at the same time, we have to remember that this oogonium is sessile. That is, this oogonium sessile. What is the meaning of sessile? They doesn't have any stalk-like structure. However, during the process of transverse division, one stalk cell would have been formed, but they are not the true stalk of this particular oogonial cell. They are sessile. So they have been embedded inside the wall of this particular cavity. They are found inside. All this procedure is happening inside this concept, inside the wall of this conceptacles. Remember. So this nucleus of this oogonial cell undergoes meiotic division, results in the formation of 4 haploid nucleus, and followed by 1 mitotic division. So 8 nucleus have been produced. And at the same time, the oogonium will also develop a thick wall around it. Oogonium is developing thick wall around it, where it is made up of 3 wall layer, that is exocyte. Outer wall is called as exocyte. Middle wall is called as mesocyte. Inner wall is called as endocyte. Okay, so now at this particular, where all this is happening inside, this oogonium has been embedded here only. It has not come onto the surface. Remember that. So this nucleus has undergone meiotic division followed by mitotic division. 8 nucleus have been formed, and at the same time, the oogonium is secreting a wall, 3 wall around it: exocyte, mesocyte, and endocyte. So later, what happens to these 8 nucleus? Only one of the nucleus will become functional, and all other 7 nucleus will degenerate. They just degenerate themselves. Only one of the nucleus will be functional. At that particular time, once all this oogonium, the process of oogonium development is happening, at that particular time, how actually, see how the conceptacle would have also been developed. So here, this is the transverse section of the developed conceptacle, where you can see this is the receptacle which we have taken the transverse section, and if it has been observed under the microscope, in this receptacular branches, there forms the cavities. These cavities are called as conceptacles, and the wall which is underlaying this cavity is called as conceptacle wall. This is called as conceptacle wall. And you can find some sterile hair-like structures. Those are called as paraphysis. And this cavity has got an opening. That opening is called as osteoles. They are called as osteole. The opening is called as osteole. Once the oogonium has been developed, this is how it looks. So later what happens once the oogonium has been developed? So here, as I was mentioning, this oogonium has got 3 wall layer. That is exocyte, mesocyte, and endocyte. And also I said this particular oogonium is sessile. Sessile in the sense they may not have a stalk-like structure at the beginning. Later, what happens to this oogonium? The exocyte of the oogonium ruptures. Exocyte, that is the outermost layer of the oogonium, ruptures, and the mesocyte will produces the gelatinous stalk. The mesocyte produces the gelatinous stalk, and this gelatinous stalk is being projected outside. This oogonium is being projected outside the osteole. Because of this mucilaginous stalk, so that once this oogonium has been projected outside, it is easy for the process of fertilization. That is the reason the mucilaginous stalk. How this mucilaginous stalk has been produced? This is due to the mesocyte, which forms the mucilaginous stalk. Okay, so how this mucilaginous stalk has been produced? The mesocyte produces the upper region of this particular oogonium. Say for example here, the developing oogonium here, what happens? The mesocyte, the exocyte ruptures. The mesocyte produces a germ tube-like structure. Mucilaginous papillae it produces. The mucilaginous papillae, it grows until it go and attach onto the wall of the cavity. The mucilaginous papillae go and attach onto the wall of this cavity and pull this oogonium up. It will pull the oogonium up. So the mucilaginous stalk will attach onto this wall of this particular conceptacle, and the oogonium is being pulled out. So once it has been pulled out, the oogonium is being exposed outside this ostiole. So now this oogonium is ready for the process of fertilization. So this oogonium has now it is called as a egg, where it has got a nucleus that is called as egg nucleus, and it has got a gelatinous stalk. The main function of this gelatinous stalk is to expose this oogonium outside the ostiole, so that it is easy for the process of fertilization. Now, coming to the development of Antheridium. So how actually this Antheridium develops? So this Antheridium has been developed as I was mentioning, from a special structure called as male conceptacles. See, this is how the male conceptacle looks. So here also, from the receptacle branch, there are some cavities. At the wall of this cavity, some of the cells gets distinguished. Those are called as antheridial initials. Those are called as antheridial initials, where this antheridial initials, they have a distinct nucleus, dense cytoplasm they have got. And these cells undergoes transverse division, where the upper cells develop into the antheridial cell. It develop into antheridial cell, and the lower cell develop into stalk cell. Thus, again, this particular sargassum, it is diploid. Hence, the nucleus undergoes one mitotic division and one meiotic division followed by several mitotic division, so that 64 nucleus have been formed in each antheridium. 64 nucleus have been formed in each antheridium. At the same time, this stalk cells also germinates, also start developing, and they can also produce one more antheridium. And if that particular branch is not producing antheridium, they are going to develop into the sterile paraphysis. Okay, once again, I will repeat. At the inner wall of the cavities or the conceptacles, some some cells become distinct. Those are called as antheridial initials. These antheridial initials undergoes one transverse division to form upper cell and the basal cell. The upper cell develops into the antheridial cell. The lower cell develops into the stalk cell. This stalk cell can again divide, and it can produce one more antheridium, or if it is not producing antheridium, those cells are called as sterile cells, where those sterile cells develops into a structure called as sterile paraphysis. So coming to the antheridium, the nucleus of this antheridium, which is diploid, it undergoes one meiotic division followed by many mitotic division. That is 64 nucleus have been formed. That is, in each antheridium, you could expect 64 antherozoids. That is, 64 male gametes are produced in each antheridium. Again, this antheridium has got two wall layer, exocyte, and it has got two wall layers. Remember, that is exocyte and endocyte. And this antherozoids, if you see the antherozoids, it is single celled and has got a single haploid nucleus, and it has got a pointed anterior region. This is anterior and this is posterior. How this anterior region it is pointed, anterior region is pointed, and it has got a lateral flagella. Two unequal flagella can be found. One is of tensile, other one is of whiplash kind. So if you see the transverse section of this male conceptacle, even this is a flask-like structure, and at the wall of this conceptacles, you can find the antheridium, where antheridium has got two wall layer, exocyte, endocyte. Inside this antheridium, you could expect 64 antherozoids, and there are certain hair-like structures, sterile hair-like structures. Those are called as paraphysis, and it has got an opening called as ostiole. Once this antheridium matures, matures, what happens? This Antheridium gets detached from that of a sterile paraphysis, and they go and land, or they get accumulated near this ostiole, and even this exocyte of this Antheridium secretes some mucilaginous structures. These once this mucilaginous structures have been produced, once this Antheridium ruptures, produces mucilaginous structures, and later this anthrozoids escape from this particular male conceptacles. Once it escapes from that of a male conceptacles, what happen? The fertilization should happen. So I have